Imagine playing Tetris, but this time with protein molecules, peptides. That is what Dr. Sridhar, Venkat, Anand, and Amit are doing at Peptris. I want to welcome Dr. Sridhar, a renowned pharma researcher from India. His, drug for tuberculosis just got approved for phase 2. Venkat, brilliant computational scientist, and Anand, another brilliant computational scientist.
Perfect. Okay. It's very hard to be on the stage after a presentation like what we saw just now. I think the next set of slides that I'm going to have, or rather the pictures that I'm going to have, are probably going to be a lot mundane. But let me at least try to make the talk more interesting. Okay. So we are the new age drug hunters. Yeah? So we see some old faces here, but we are the new age drug hunters. We are here to, thank you. We are trying to solve some of the most complex problems facing drug discovery today. And I think Vish made a statement about he using ChatGPT a lot. So I also do a lot of rephrasing with ChatGPT. And some of the content that you see here is coming from, what do you call, a rephrased content. So they are my quotes, but helped by ChatGPT. And we are a very complex system. We are a product of evolution over billions of years. And what we are currently, if you really think about it very philosophically, a complex chemical reaction that is going on. Multitudes of chemicals interacting with each other, and that is what we are. And it's in perfect balance, and that is why we're able to walk, talk, think, speak, and so on. So I would say what we sense and how we make sense of it, all of it is a chemical reaction. And any imbalance in it, or any runaway reaction in the chemical reaction within, causes diseases or whatever, some abnormalities and so on. And here we are trying to figure out how to keep it in this pristine form, as in rather at least in this form, to ensure that we all live in good health. And when we talk about this perfect balance and the imbalance there, trying to get it back into balance is what the biologists are trying to do always, to ensure that we have good health. And if we try to, and we make it for a much longer period, then that is what longevity is about. Being able to live longer, healthier. And I have to actually start the story with some of these things that we know about biology. We really hardly know anything about our human body per se now. Biologists are still trying to figure out what is happening within our system with all these complex reactions that are happening and their interconnections and how does it get out of balance and so on. They are actually being the detectives in this world of trying to understand each and every cell and how it interacts and so on. And here comes the final identification of a target, as in all these things. When I'm looking at it as an outsider, not as a biologist, as an engineer, I look at it as a complex control system. There are too many protein knobs that they are looking at, and drug discovery started off with randomly tuning all of these knobs and seeing whether anything makes any sense, changing them and so on. And now there are probably AI is helping there to figure out how to find out what are the right targets to go behind. And then comes the chemistry part of it. The chemistry is pretty much simple. So now you know how to tune a particular sensor or a signal. That is, these are all proteins. The chemistry is quite simple. There are just about 10 power 60 molecules. You need to pick one of them. It is like picking a needle from a haystack and figure out how it can modulate the particular protein of interest that we have. The challenge is not about modulating that particular protein target the biologists have found. The biologists also have a condition saying that you shouldn't modulate anything else. Because as we all know, having drugs leads to side effects, and most of the side effects are because of the drugs that we are taking, or most of the conditions that we end up in. And how do we ensure that we modulate these targets without affecting anything else? There should be no off-target effects. And believe me, it's very simple, because there are just about 20,000 proteins that we should not be interacting with, other than the one that we need to interact. And the good part is we don't know anything about most of them. Yeah, about a good 90% of them. So this makes it very challenging, and that is why drug discovery is really hard and very complex and leads to very late stage failures. So before I pass it on to Sridhar, I will just give you the larger landscape as to what AI is doing in the healthcare. So talking about all these predictions about finding the right target and modulating them and all, we also need to look at AI is helping us with our longevity, right from prevention, prediction, finding out the therapeutics and well-being. So we used to have the saying that an apple a day keeps the doctor away. Very soon it could be that the apple may be replaced with an Apple Watch, because the amount of data that it is collecting and helping you predict failures and so on, predict like any health issues and so on, is going to be a lot faster, and getting you treated where there is a lot of AI again, yeah, in terms of so to say the biomarkers that we are finding out, the diagnostics that we are doing and so on, everything, and right from mental health to mobility, everywhere AI is all over there. Over to you, Sridhar.
Thanks, Venkat. I like to make my presentations very interactive. So I'd like to show of hands how many biologists in the room. I've got one, Shweta there.
Some there.
One more. Yeah, about a handful of them, right? So I'm going to make this as simple as possible, right? And that's how I trapped these three gentlemen, Anand, Venkat, and our colleague there who chooses to be different and stay at the table there, Amit. I explain biology in a very simple way to get people to understand very complex problems. So I'm going to try to explain to you what drug discovery is, a journey of about 5 to 7 years in about 2 minutes, right? So bear with me. So this is how traditional drug discovery used to work. Most of the big pharma used to follow this approach at least 5 to 7 years ago, before the advent of AI into the pharma space in its true sense, right? So if you can see, oops, sorry, do I have a pointer here? No. No, right, sorry. You can see a key under the traditional drug discovery there, right? So I generally use a concept of a lock and key when I explain how a protein interacts with a chemical, right? And it's probably the simplest way. So Venkat mentioned 10 raised to the power 60 chemical molecules, right? So imagine a bunch of keys with 10 to the power 60 keys in it, right? And you have one lock to pick, which is the protein of interest. And you have several other locks, which are those 20,000 proteins that you don't want to hit, right? Imagine how difficult it is going to be to pick that one key and fit it right in, right? And that's what we have been doing over the years. All the drugs that you take to treat some of the different diseases have all come through the traditional method of discovery, where you have a disease hypothesis. So everything starts with biology and ends with biology. There's a lot of chemistry and other things in between, right? So you have a disease hypothesis. Let's say, for example, diabetes. And you say, how am I going to treat diabetes? There are so many ways in which you can treat diabetes. And every company works on its own different way of treating the disease. So that's the hypothesis and identification of a protein target which you want to work on. You then go to the drug molecule, which is the chemistry space, and see which one of them interacts with these targets, right? And once you have identified that, you start the basic tests of manufacturing that molecule, optimizing it for drug-like properties, and then testing it through various cell-based systems and animal systems. One of the things that most pharma companies will follow is fail fast, fail cheap, right? So all of these process is to make sure that you have a molecule which can actually become a drug, right? And any time you fail is cost that has gone down the drain because you don't have anything tangible to make out of it. So we reached the stage of animal testing. So what we do at Peptris is work up to this stage of animal testing where we can detect failures at a much earlier rate and also at a much cheaper rate. After this, it goes into human clinical trials and then into regulatory approval. The total duration of this entire thing is roughly about 12 to 15 years total, of which the preclinical part is about 5 to 6 years. And the total cost of investment in this preclinical part is about $670 million, right? So once you're spending so much of money and you're failing, all of that money is lost. What we do at Peptris is try to bring in efficiencies in this whole process. Anand, over to you.
Thank you, Sridhar. So, as Sridhar explained, if you look at the pipeline, where we concentrated is where can we apply the engineering principles so that most of the uncertainty that we see in the whole pipeline can be avoided and can be automated. And we fixed in the first preclinical stage where trying to find a molecule or a key to a given target or a protein is what we think we can apply our engineering principles. And that's where we applied. The 4 to 5 years that Sridhar explained, we actually shrink it down to approximately to 18 months. And how we do it? We actually built a suite of algorithms because most of these interactions or most of these predictions are so complex that one algorithm does not suffice it. So we actually built a suite. When I look at the amount of the keys that we have, say 10 raised to 60, how do we really imagine searching about such a huge one? It is intractable for even a computer to iterate or enumerate any of them. So what we built is a very unique molecule representation, which in the first stage try to distill this large library into something that works for this protein. And once you identify a short list, we push it through even finer and higher accuracy predictions into a full interactions with that protein and trying to understand how really it works. Third, which is also key for a drug discovery, is about whether this molecule has any of those properties that make it like a drug. Will I be able to ingest it in my intestine? Will it get into my blood? Will it go to a real organs that it is required? All those properties can actually be predicted, which in turn helps to avoid late stage failures. If we know what are the problems, you can actually optimize it even in the later stage. And finally, the third, which is also a very important part of here in this, the generative AI that helps us to generate novel molecules that have not been synthesized, that have not been seen among the universe of 10 to 63, but an algorithm can actually explore them and give you a solution that will really exist and can be synthesized. With this, we finally do a wet lab experiment to confirm all our predictions, and now we have a lead molecule that actually can get into a drug. It has to go through all other rest of the pipeline, but it is as quick as possible that within 18 months, we're able to identify a lead molecule that can be taken further. Now, with that, what we achieved till now with the Peptris is we focus on 3 areas: the oncology, inflammation, and the rare diseases. We took a problem set in each of these domains, and we started applying these algorithms. And we actually have a very good progress in a rare disease, wherein we have 2 molecules that have come out to a lead optimization stage. By this, what we mean is these 2 molecules work for this disease, have shown some benefit in the experiment that we have conducted in the lab and also in the animals. Predominantly being repurposable drugs, it means they're already FDA approved, they're already safe. They can actually get into clinical trials very quickly and help the patients which are in need of it. Now, to look at the advantages of the Peptris, in the 2 case studies that I'm going to present here, the first one is a rare disease. Among all the list of 300 million molecules that are available, we shortlisted just 33 of it. We did an experiment just buying 33, and we got 6 active molecules, 6 molecules that actually work on our hypothesis. And this is coming from an actual wet lab experiment. We look at a repurposable list, we identified one, and it is also an active molecule. And in fact, they show much better functional effect than any other known drug till now for that particular rare disease. Similarly, on a very different inflammation target or a different disease, we identify 62 targets, 62 molecules. And when you test them, we see that around 30 of them are active. So this kind of enrichment that you see, 60 being predicted, coming somewhere between 10 to 30, around 15 to 50% activity that within a molecule is unheard of. Because it gives me a power to distill 300 million molecules and come out with a drug solution. I don't need to test all of them in the lab. Rather, we need to test very little, do it as quickly as possible, and get to the next stages.
So basically, in the last slide, I just have to also highlight for some of the non-biologists and the non-pharma background. A typical pharma company, yeah, if they have a 1 million compound library, test it, and then get a few handful of hits, they're going to be super excited, saying that, oh, their program is already started, and they're going to do something, they're going to be able to develop it forward. And here we are, AI ushering in the kind of, changes to what is happening in this landscape. And I'm sure that we are going to kind of speed up and make it much, much faster, much cheaper to get a drug to the market. Of course, thanks to Speciale. And I think, of course, from what we have been saying, we have been in this journey for the last 4 years, and we have met quite a few VCs on the way. And most of the time, understanding, before they even understand the concept, they jump into when will you make money, what will you be doing beyond, and so on. The questions is your timeline going to be, will you be in revenues in 1 year? Will you be revenues in 2 years? What is your current revenue rate, and so on. So these are questions that an early stage drug discovery company just cannot answer. We know that we can talk about the science that we are doing, and we know that we can tell the destination that we want to reach. But there are going to be, of course, a lot of surprises on the way. But we know that we back ourselves to solve some of the toughest challenges that come our way. So we really wanted groups, VCs, who believe that we'll be able to overcome those challenges on the way, who understand the space and know that the time that is required to get to the destination is going to be rough and it is going to be long. Sridhar, you want to add something here?
No, I completely sync with what Subbu mentioned. Vishesh and Vishnu don't know what they are getting into. This is a long journey, right? But we are very glad that they want to join the journey with us, and we look forward to many of you being part of it as well. Hopefully we can make an absolute change in the way we treat patients and bring about a change in the healthcare systems. Thank you so much.